Okay, in 9.2 we are looking at how we can copy DNA without having the cell needing to do it for us. So that is what we are going to be examining in this section. So PCR, which stands for polymerase chain reaction, uses polymerase to copy DNA segments. PCR can make many copies of specific DNA sequences in just a few hours. So it can be done with if you find pieces of DNA at a crime scene, or you're wanting to get a sample of DNA from like a woolly mammoth or something like that, you can remake a whole bunch of that DNA. So PCR amplifies the DNA samples, and PCR is similar to DNA replication. PCR is a three-step process. PCR uses four materials: the DNA to be copied, DNA polymerase, the nucleotides adenine, thymine, cytosine, and guanine, and two primers. Those are going to be the four things that are needed for this whole process. And there's really three steps of PCR for it to really work. So the first thing that's going to be done is you're going to use heat to separate the double-strand DNA molecules. So that heat is going to cause the DNA to separate from one another. You will take the primers that you have and you're going to bind it to each of the DNA strands at the opposite ends of each other. And then you're going to use the DNA polymerase to bind the nucleotides together to form the new strands. So that's what you see occurring down here. So the DNA strands right here are starting to separate. Then you add the primers, which there's one primer and there's another primer right there. You add those, and then the DNA polymerase is going to start building the nucleotides in between. And again, you're going to end up with an old strand paired with a new strand. This process can continue on and on as long as you have the materials that you need. So each PCR cycle doubles the number of DNA molecules. So you can see within a matter of like a couple of hours, you could have billions of copies of the DNA that you're wanting to work with made. This can be used in DNA fingerprinting. It can be used just in regular genetics, any genetic engineering things. You need more of that copy of DNA. You just make more and more copies of it. So we're going to look now at the DNA fingerprinting. Like I said, you can use PCR, and you will use PCR when you do DNA fingerprinting as well. So a DNA fingerprint is a type of restriction map. DNA fingerprints are based on parts of individuals' DNA that can be used to identify them. So it's going to be based on non-coding regions of DNA. So non-coding regions have repeating DNA sequences. Number of repeats differ between people. So when you have those differing numbers of repeats, you're going to end up with different banding patterns that you'll see in the electrophoresis gel, and that's what we refer to as a DNA fingerprint. So that's what you see representation of it down there on the screen. So if you use the same restriction enzymes and cut it at the same spots, if you're wanting to see who committed a crime, or is this person's DNA, if you're using the exact restriction enzymes, it's going to cut it at the same spots if it's the same person. DNA fingerprinting is used for identification. It depends on the probability of a match. Many people have the same number of repeats in certain regions of DNA. The probability that two people share identical numbers of repeats in several locations is very small. So They use it to look at, well, or these people's kids. You'd be able to see, oh, there's a lot of similarities between their repeat sections, and so yes, you are like this percent chance of being that kid's parent. So that is how they end up using DNA fingerprinting for that. Individual probabilities are multiplied to find the overall probability of two DNA fingerprints randomly matching. So 1 out of 500 times 1 out of 90 times 1 out of 120 equals 1 out of 5.4 million people chance for a DNA segment, for example. So several regions of DNA are used to make DNA fingerprints. So DNA fingerprinting is used in several ways. Evidence in criminal cases. We will be doing a lab where you're going to be doing a paper version of a DNA fingerprint. It can be used in paternity tests, immigration requests, saying like these people are related to one another, so they have to go together. Studying biodiversity. That turtle right there is going, oh, that tickles, stop. So that is some things, tracking genetically modified crops. So that is the end of the notes for section 2 and 3.